Hello, everyone, and welcome to today's webinar, Beyond SARS-CoV-2, Application of Absolute Q Digital PCR System for Waterborne Pathogens. I am Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, please visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Huiyun Wu, research scientist at Tulane University, School of Public Health and Tropical Medicine. For a complete biography of today's speaker, click their name in the presenter's window at the top right of your screen. Dr. Wu, you may now begin your presentation.
Hello, everyone. My name is Huiyun Wu. I'm a research scientist at Tulane University, the School of Public Health and Tropical Medicine. I'm also from the Department of Global Environmental Health. My research area is on water microbiology and virology. Specifically, it's on microbial source tracking, wastewater-based epidemiology, and I study the fate and transport of water pathogens and the microbial contamination indicators. I've been using the qPCR such as QS3 and the STEP+1 system in the past intensively to detect waterborne pathogens, and I started using the Absolute Q system since I joined Tulane in April this year. It is an honor to be invited by Thermo Fisher today to share my user experience of the Absolute Q digital PCR system for water and environmental health research projects we have been working on in Tulane University. Water is important to the public health. One of the goals is to make sure the water surrounding us is drinkable, swimmable, and fishable. Therefore, we need to monitor and study the waterborne pathogens and the diseases caused by the waterborne pathogens. There are a few groups of waterborne pathogens in the environment. They can be bacteria, such as Salmonella and E. coli. They can be viruses, such as the hepatitis A norovirus, poliovirus, adenovirus, and they can be protozoa, such as cryptosporidium. These pathogens are usually transmitted from the fecal-oral route, since they can be excreted in high numbers from feces. When the water body is contaminated by feces or sewage, the pathogens can be transported in water and cause waterborne diseases. These pathogens can be resistant to disinfection approaches, and some of them can survive in water environment for a long period of time. For example, crypto is resistant to UV disinfection, and E. coli can propagate itself in the environment when the temperature is appropriate. Some of the waterborne pathogens are zoonotic, which allows them to break the boundaries between animal and human and transmit across the species. The U.S. EPA Environmental Protection Agency has proposed a microbial contamination candidate list, and several microorganisms are listed in the table, including adenovirus, norovirus, Salmonella, Mycobacterium avium, and so on. These pathogens are selected because of the diseases and infections they are causing, and they might be regulated by the federal government in the future. The most common disease caused by waterborne pathogens is gastroenteritis. Besides this disease, adenovirus and enterovirus can cause respiratory illness. Mycobacterium avium and Legionella can cause lung infection. Polio can cause meningitis, and hepatitis A can cause liver disease. Among the emerging pathogens, viruses are causing 44% of the infection, followed by bacteria and protozoa. Virus can infect algae. It can also infect animals, including fish, bird, and mammals. Virus can also infect bacteria, which is also called bacteriophages. For public health concern, viruses can infect humans. These are the top viruses that affect water quality, and most of them are in the EPA microbial contaminant candidate list, such as adenovirus, hepatitis A, norovirus, and enteroviruses. It is critical to ensure water microbial quality for the public health. However, there are many challenges to detect waterborne pathogens in the environment. Some of the waterborne pathogens, such as the viruses, has low infection dose. People can get sick when they are exposed to a few pathogen particles, especially when they are young, old, or immune compromised. The pathogens have low abundance in environmental waters. Therefore, it is not easy to detect them directly, and it is always usually require us to concentrate the water samples for the pathogen detection. However, when we are concentrating the water samples, the inhibitors in the environmental samples can be also concentrated at the same time. The inhibitors will challenge the molecular target detection in the PCR and qPCR reaction. Therefore, to study the waterborne pathogens, we would need to navigate the pathways to conquer these challenges. First, I would like to introduce you some water processing approaches for pathogen detection in our lab. The membrane filtration is the most commonly used method to process water samples. Water samples will go through the membrane filter, and the particles and pathogens that are greater than the membrane pore size will be kept on the filter for downstream analysis, such as extraction and PCR reaction. An application of the membrane filtration method is to filtrate 100 milliliter of lake water sample using the 0.45 micrometer pore size filter for bacterial, pathogen, or fecal indicator detection. InnovaPrep ultrafiltration system is a novel water processing method for waterborne pathogens. In our lab, we use it to concentrate 200 milliliter lake water samples for virus pathogens. The water samples will go through the pipette tips, and then the pipette tips will be eluted for the concentrated water samples, and then the concentrated samples will be ready for extraction. For larger volume water samples, we use the hollow fiber ultrafiltration approach, which is also known as the HFUF method, because this filter column here, it has a larger surface area. In our lab, we process 20 liters of the secondary effluent through the hollow fiber filter using a peristaltic pump, and then we would elute the filter with the elution buffer using the backwash approach, and then the elution buffer contains the phosphate and the twin-20. Nanotrap hydrogel particles is another novel approach to concentrate the water samples. In our lab, we process 35 milliliter untreated wastewater samples with the hydrogel particles. The particles will capture the viruses based on its membrane affinity to the viruses. And then the viruses will be released after wash, and then it'll be ready for extraction. We use this method to detect the monkeypox virus and SARS-CoV-2 virus from untreated wastewater samples and the manhole water samples. After the samples are concentrated, we perform extraction for the nucleic acid and prepare the pathogen detection on digital PCR. Here I would like to show you the procedures that we are performing in the lab for the digital PCR preparation. The absolute Q system is similar to regular qPCR systems such as Step +1 and QuantStudio 3 in terms of the reagent preparation. I always prepare the plate outline before I start the experiment. Here's an example of detecting enterovirus from particles in secondary effluent. Secondary effluent is a type of treated wastewater before final disinfection and before it's released to the environment. We ordered the primers and probe from a commercial vendor in a dry format and dilute them with tea buffer. As you can see in the table, the stock concentration is 500 micromole for primers and 100 micromole for probe. Since digital PCR requires higher concentration of primers than the probe, we use 1 micromole for forward and reverse primers and 0.25 micromole for the probes in digital PCR reactions. I usually prepare 200 reactions at one time and make 10 aliquots. There are 20 reactions in one aliquot, which is sufficient for a 16 reaction plate. I prepare the primers and the probe PP mix aliquots ahead of time so that I can store one aliquot for one plate. Even though digital PCR provides absolute quantification, we still run the samples in duplicate, as you can see in the plate outline, to double confirm the quantification. And we always include a positive control and a negative control in the same plate with the samples. Enterovirus is an RNA virus, and in one reaction mix, we load 0.5 microliter of the prepared PP mix and then 2.5 microliter of the one-step digital PCR master mix, which is 4 times concentrated from Thermo Fisher. And we also load 2 microliter of water and 5 microliter of the RNA template. So in total, we prepare the reaction mix in 10 microliter. Here's another plate outline to detect monkeypox virus as an example of the double-stranded DNA virus. Since monkeypox is a DNA virus, we use the dPCR master mix that is 5 times concentrated. And the other steps are similar to the previous plate outline. We use the same working concentration for the primers and the probe, and we prepare the PP mix in the same volume. These procedures are to ensure that the results are reproducible and reliable. After the 10 microliter reaction mix is prepared, we load 9 microliter into the MAP plate, and then we load 15 microliter of the isolation buffer on top of the reaction mix. We then cover the plate gasket strips on all of the 5 columns and load the plate into the absolute Q instrument. The entire qPCR preparation can be done in 30 minutes when the lab personnel is proficient and then fully trained. And the qPCR reaction can be done in 2 hours, which is beneficial if the sample requires fast turnaround results. Here I'm going to share the quality control acceptance criteria that we apply in the lab. In general, a sample is considered positive when there are more than 3 microreaction chambers are positive. We set different fluorescence thresholds for different dyes. When FAM is used, the fluorescent baseline is set for 10,000. When hex of Sci5 is used, we set it for 1,000. When VIC is used, it's set for 2,000. As I mentioned before, digital PCR require higher concentration of the probe. So when the probe don't have the sufficient concentration, 0.25 micromole, or when the probes went through multiple free source cycles, it can lead to some abnormal fluorescent levels in the separation chart, which requires further troubleshooting. We expect to see all of the negative controls shows less than 3 positive microreaction chamber, including the non-template control, processing negative control, extraction negative control. In these controls, if these controls shows more than 3 positive microreaction chambers, it is a sign of contamination, and we need to do some troubleshooting or rerun the plate. We expect to see the total number of the reaction chamber in one well is greater than 20,000, and we expect to see the positive control shows greater than 3 positive microreaction chambers. For environmental samples, as I mentioned before, the concentration might be very low. So the criteria we are using in the lab is we consider that a sample is positive when there are more than 3 positive microchambers that are detected, and then more than 20,000 total reactions are detected. All of the negative controls are non-detected. All of the positive controls are detected, and there's a clear separation in the dot graph. If one of the items did not pass, we will need to rerun the samples. So for the digital PCR provide us absolute quantification, and I would like to show you some examples of the samples that we completed in the lab. I tested some virus pathogens positive controls on absolute Q. I extracted the DNA from human adenovirus, 40 virus stock for positive control. We purchased G-block standard for enterovirus, Coxsackie B, norovirus G2, hepatitis A for positive control. They all showed very clear separation. The blue dots blue dots are fan dye, and we set the threshold for 10K. The green dots are weak dye, and we set the threshold for 2000. I also tested some bacterial pathogen positive controls on absolute Q. These are the separation charts for Campylobacter and Salmonella G block standards. These are two pathogens in the EPA CCL list, and then they are quantified with different fluorescent dyes. As you can see, they are in different colors. By using different dyes, this can provide a potential to run them in duplex in the digital PCR system in the future. I also tested some virus fecal indicators, positive controls on digital PCR. They are BX174 and MS2. BX174 is a single-stranded DNA virus, and MS2 is RNA virus. They are both bacteriophages. The positive controls are nucleic acid extract from the virus stock. The absolute Q system enables us to do multiplex plates, which allows running several targets from the same sample on the same run. Here, I would like to show you some examples we completed in the lab. In this plate, we tested the HF183 IAC assay for the standard. The HF183 is a human fecal indicator. It is a human host-specific bacteroides. Recently, the US EPA has drafted a method 1696 for its application to quantify fecal contamination from human waste in environmental water samples. The method proposed an internal amplification control, which is the IAC here, so that the PCR inhibitor can be identified at the same time the fecal contamination levels is quantified, which is a duplex. We applied this assay in absolute Q system successfully, and here is the separation chart. We used the SAM to quantify the fecal contamination level, and the weak, which is the green dots here, to identify the inhibition level. The internal amplification control plasmid is spiked in the reagent mix. So if the samples are not inhibited, the weak quantifications should be similar to NTC. And in this way, we can get both absolute quantification results of fecal contamination and the inhibition at the same time. We also applied the SARS-CoV-2 commercial kit on the raw wastewater samples that we extracted directly. The kit has 3 targets inside, and we can quantify SARS-CoV-2 M1 gene, M2 gene, and PPMOV gene together. The PMOV is a plant virus, and it is found to be the most abundant virus, RNA virus in human feces. Here it is used to quantify for the quality assurance in SARS-CoV-2 assay from wastewater. Besides the absolute quantification, the absolute Q system can provide a sensitive quantification, and I'm going to show you some examples that we have completed in the lab. In this project, we are trying to find if there are virus pathogens associated with the particles in secondary effluent for water reuse purposes. Since the secondary effluent is treated wastewater, the concentration of the pathogens might be too low to detect. The particle-associated virus is a portion of the total virus, so it might be even more challenging for the detection. In this experiment, we use 3 liters of the secondary effluent samples, and then we process it by the membrane filters with different pore size. On the digital PCR, we were able to find a positive well with 0.5 gene copy per reaction. And then we factored in the qPCR preparation volume, the nucleic acid volume that we load in the reaction, which is the 5 microliter, and then the extraction volume and sample processing volume for the back calculation. After we do the back calculation, we found that the concentration of the human adenovirus associated with this particle is 60 copies per liter. We also detected the norovirus from the secondary effluent particle samples as well for the final concentration of 272 copies per liter. And also we found enterovirus from the secondary effluent particle samples. The sample 7.3 has 3 positive microreaction chambers, and it is quantified as 0.34 copies per microliter. As I mentioned in the QC criteria, we can still consider this sample is positive since it's greater than 3 positive microchambers. And then after back calculation, we get the final concentration of 45 copies per liter, which is the detection limit of the sample processing method that is combined with the digital PCR. We detected monkeypox from raw wastewater samples. So in this project, we processed 35 milliliter raw wastewater samples using the nanotrap hydrogel particles. And after back calculation, we found that the concentration of monkeypox in one sample is 3,100 copies per liter. And then the final concentration is greatly influenced by the concentration factors and the sample processing method we are using. Above all, is my experience using the Absolute Q digital PCR system. We can get the absolute quantification of the waterborne pathogens without developing a standard curve. And then the Absolute Q system has similar procedures to prepare the reagent mix compared to qPCR. So, and then the experiment can be completed in 2 hours. The detection limit is as low as 0.34 copies per microliter per reaction, which corresponding to 1,500 copies per liter or 45 copies per liter in environmental water samples, depending on the concentration factor. We could also use the digital PCR to evaluate PCR inhibitors for the samples. Finally, I would like to acknowledge the support my team has provided to the project in this presentation. And then the research is funded by EPA STAR grant and by US Navy. Thank you so much for your attention, and I'm happy to answer your questions now.
Thank you, Dr. Wu, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. and we'll answer as many questions as we have time for. Now let's get started. Our first question is, how did you determine where to set your threshold values?
Thank you so much for your attention and for your question. So as I mentioned in the presentation, we have choose several different dyes to detect the pathogens. This is also allow us to be able to do duplicates or multiplex in the future. And then because different dyes have different fluorescent signals, this is required us to do some positive runs and negative runs to determine the threshold level. Basically, we would compare the positive signals and then see where the separation appears, and then compare with the negative wells. So for FAM, in our lab, we see it is consistently showing the separation around the 10,000 K signals. So we stay with 10K for the FAM dyes that we use for the pathogen detection, and it has been working pretty well in our lab. But when people are using a new assay, it is also recommended to do the same procedure, compare the positive control and negative control, and then determine the threshold level and stay with the same threshold level for the same target. Yeah, and then the same thing applies to other fluorescent dyes.
Great, thank you for that. Another question here, what is the dead volume for the reaction?
So in the absolute Q manual, it is mentioned that the dead volume is 5%. So that means like over 95% of the reaction mix that we load in the plate is used for quantification. However, in our lab, we only have one digital PCR system, and we cannot compare across the platform, and we don't have super significant concerns of the reagent that we loaded into the plate that is being used. So I would stay with this less than 5% number for now, and then I think if we see some differences in different platforms, we can do further investigation and then comparison.
Thank you. Some good questions coming in here. The next one is, were all of the concentration methods comparable, or was one better than the other?
So this is a very good question. We study environmental samples, especially the water samples in our lab. And depending on the types of water samples and the types of basically depends on the characteristics of the samples, we would have to choose different concentration method. As I mentioned, the membrane filtration is the most commonly used one because of the cost, the level of training we need to provide to the new fellows in the lab. However, there's also very limited concentration factors. So therefore, we have to adopt different method for different target. That's why we introduced the InnovaPrep system to detect the viruses. And then we also we were trying to use the hydrogel beads for the raw wastewater samples to capture the viruses and other pathogens in the matrix. So I guess there's no short answer for this one. It depends on the concentration factor you are looking for. It depends on the samples you are working with. Yes, thank you.
Great, thank you. The next question here is, while dPCR is more resistant to the accumulation of inhibitors, do you think the additional cost and reduced well count associated with the technique makes it a better option over qPCR?
So I think from my experience, the reason that digital PCR is more resistant to inhibitors is because of its low sensitivity, high sensitivity, low detection limit, which means the samples can be diluted more and still be able to detect it. I still need to do more comparisons with the inhibition levels using the HF183. Like I mentioned in the presentation, we have established the assay in the digital PCR, but we still need to use this assay to quantify the inhibition level for the samples. And also, I think in terms of the cost, as more and more people are using the digital PCR platform, the cost will reduce in the future, like the sequencing nowadays for us. So I guess the cost will be comparable eventually in the future with the qPCR system. The only concern I have besides the cost is the throughput. But I think people are trying to increase the throughput of the digital PCR so that we can do the quantification in a larger batch.
Great, thank you. We have time for a few more questions. Someone said, thank you for a beautiful presentation, and is interested in the virus recovery method used, if you can speak a little bit about that.
Yeah, thank you. So we have tried different concentration method, and we found that the InnovaPrep and the nanotrap hydrogel beads, they both have around 20% of the recovery in terms of the virus pathogens. We did that by comparing the direct extraction and the concentrated pathogens. So I guess my answer is around 20% is what we found from the samples.
Thank you. Now we'll wrap up with this question. Why did you choose 5 microliters of DNA or RNA for the reactions?
Yeah, so for the digital PCR that we are using here in Tulane, we are loading 9 microliter in the wells, but we prepare 10 microliter for the reaction mix. And then out of it, we use 5 microliter. The reason we use 5 microliter is the pathogens are usually hard to detect. So on one side, the more DNA or RNA you load in the reaction mix, the higher chance you will get the pathogens. But the other side is since the samples are usually containing the inhibitors, and after the concentration method, the inhibitors are also accumulating. So the more samples we add, the more inhibitors we add as well. So we chose 5 microliter as the sweet zone of the balance from the detection of the pathogen and then the consideration of inhibitors in the samples. However, if you look back to my equation or the plate outline, for DNA target, we added 2 microliter of the digital PCR master mix and 0.5 microliter of the PP mix, which gives room for 7.5 microliter of the DNA sample. So you can still play around in the system to choose different numbers for your result. And then as long as we can get the sweet zone that we are satisfied, I guess you can choose different volume as well.
Great, thank you. And thank you again, Dr. Wu, for your time today and your important research. We'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. And before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we do not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, goodbye.
Bye.